So for those of you who don't know, Nohla is a private cell therapy company based in Seattle, Washington. We have about 50 employees, and we're focused on the development of off-the-shelf universal cellular immunotherapies, all designed to address patients with critical diseases. So we have a novel platform. We are a platform company. We leverage the Notch signaling pathway to direct the differentiation of CD34 cells that are isolated from umbilical cord blood. And our initial application is in patients with hematologic malignancies. These are patients that are high risk for morbidity and mortality associated with the treatments they're receiving. In the near term, we have data that will read out in allogeneic transplant, specifically in cord blood transplant. We have a randomized trial that is fully enrolled with 160 patients, and we expect to report the results of that study in the first quarter of next year. And then we've initiated a large global study in AML patients who are getting intensive chemotherapy. That study, we've enrolled about 85 patients. It's a 220-patient study, and we expect to fully enroll that in the first half of next year and report that data out by the end of the year. Based on discussions that we've had with a number of regulatory authorities over this past year, we do have signals that if the data in our phase 2b cord blood study is positive, there are routes for potential early approval in some of those territories. And this initial market opportunity is large. For those of you who are familiar with the hematologic malignancy space, in allogeneic transplant, we're looking at a market of about $2 billion, and in AML patients getting intensive chemotherapy, it's about a $1.2 billion market. This is worldwide. Some of you may be aware we recently completed a Series B round of financing that now has injected a little over $100 million into our company. That gives us a runway to complete our phase 2 studies, initiate our phase 3 program, and also support the transfer of our technology to a commercial manufacturing organization. So I mentioned we're a platform company, and what we do is we leverage the Notch signaling pathway. This is a pathway that is well conserved. It's a powerful regulator of telling hematopoietic stem cells to either self-renew or to differentiate. And I want to spend a minute with this diagram. What we do is we engineer a delta Notch ligand that attaches to a receptor on the hematopoietic stem cell. And this induces an endogenous Notch signaling path that tells those cells to either self-renew or, depending on the density of that delta ligand, those cells can differentiate into other cell lineages. And that really provides the pipeline that we have. I'm going to spend most of my time talking about dilanubicel, which is our stem and progenitor cell product. But there are other programs in discovery that are looking at other types of cell progeny, including NK cells. So if we look at patients with hematologic malignancies, we know that there's a significant need for improved treatment, since the majority of these patients, just by nature of the type of regimen they receive, are at high risk for pancytopenia and myelosuppression. And this is really a leading cause of morbidity and mortality in these patient populations. So in the intensive chemotherapy segment, these patients, nearly all of them, become pancytopenic, and about 20% of them are going to die from those types of infections. What we're looking to do with dilanubicel is to reduce those patient deaths, and as importantly, allow physicians to not interrupt the chemotherapy regimens that they're giving these patients with the intent of getting them into a remission. Because for these patients, the only curative option is a transplant. And yet, even with transplants, the side effects are daunting. And so, again, our intent is to reduce those treatment-related morbidities, reduce acute GVHD, and the time to neutrophil and platelet engraftment, so reducing overall infections. This slide gives you an idea of what this initial market opportunity looks like. You know, our initial focus is in cord blood transplant, and as I said, this phase 2b study that we'll read out in the first part of next year provide a pathway for accelerated approval within that market segment. But we're also looking at expanding into other segments in the allogeneic transplant space. Dilanubicel is a product that's added to the standard of care. And so the intent is adding dilanubicel in the haplo setting and the matched unrelated donor transplant setting, again, provides additional opportunities to improve morbidity and reduce morbidity and improve mortality in this patient population. We also have a small proof of concept study we're initiating in the autologous transplant space. This is a trial that was suggested by Sergio Giralta at Memorial Sloan, looking at adding dilanubicel in multiple myeloma patients getting an autologous transplant. And then we've talked about our initial entry into the larger intensive chemotherapy market with the initial focus on AML. So I want to spend a minute on this slide. This is a slide that walks through the treatment algorithm, in this case an AML patient. And what I want to communicate here is that dilanubicel is added to the standard of care. So there's several opportunities along this patient's treatment pathway where dilanubicel can be used. One is an induction, added when patients are getting induction chemotherapy with the intent of reducing infection. Second is in consolidation or reinduction in consolidation. And then if those patients get into a remission or you reduce the amount of minimal residual disease and they go to a transplant, there's an opportunity for them to see dilanubicel again. So commercially, you can see there's a lot of synergy in using this product within patients with hematologic malignancies. So the question is, how does this product work? And we know that dilanubicel elicits both an innate and an adaptive immune response. When dilanubicel is infused in patients who either receive intensive chemotherapy or have received a stem cell transplant, we see an initial functional wave of hematopoiesis. So we're generating an innate immune response, and you can see that up in the upper right-hand corner, where we're restoring the de novo functional cells like NK cells and neutrophils and macrophages. And these are all designed to improve the rate of infection. So minimize the delay in engraftment, reduce infections, and ultimately, hopefully, improve mortality. We're also learning, though, that we're modulating an adaptive immune response. And there are cells, early progenitor cells in dilanubicel that are giving rise to immunomodulatory cells. And we believe that these cells may be the reason why we're seeing no incidence of acute GVHD in our initial transplant studies and creating more of a tolerogenic effect of this product. So I don't have time to go through all of the specifics of the data that we've generated with dilanubicel, but this is a slide that summarizes the studies that we have done. We have seen some compelling findings. We've infused over 150 patients to date. One of our initial pilot studies in intensive chemotherapy, again, these are AML patients. This was a 29-patient study, and we looked at we did add dilanubicel to a high-intensity chemotherapy regimen. And what we saw is that in those patients that were given dilanubicel, we saw a reduction in infections, and more importantly, we saw no gram-negative rad bacteremia, so a reduction in bacteria and fungal infections and no gram-negative rad bacteremia. In cord blood transplant, we also saw, this was, again, a single-site study. This was in 15 patients. They received a cord blood transplant plus dilanubicel. And we saw a shortened time to both neutrophil and platelet engraftment, but we also saw no incidence of reported acute GVHD, and this translated into an improvement in transplant-related mortality and overall survival. And for those of you who might have been at EHA in June, we presented the long-term follow-up on this study where patients were out beyond 5 years, and we still saw this 87% disease-free survival hold up. So that data was the catalyst for the initiation of this trial in cord blood transplant. So this is the study I'm referring to that we have fully enrolled. It's a randomized trial at several sites within the U.S. looking at dilanubicel plus a cord blood transplant versus conventional cord blood transplant alone. And the primary endpoint is neutrophil engraftment, but there are a number of secondary endpoints to include survival, reduction in infection, improvement in platelet engraftment. We also have embedded a fair amount of translational research in this study to understand further how the product is working within this population. So as I said, this trial is fully enrolled, and we expect to read this out in the first quarter of next year. The second study is a large global study we initiated earlier this year in adult AML patients getting intensive chemotherapy. This is a trial, again, that's being done in the US, South Korea, and Australia. We have about 36 sites. This is a large trial, and the reason for that is because we're not only looking at how dilanubicel, when added to intensive chemotherapy, reduces the rate of infection, we're also looking to find the minimum effective dose in this patient population. So that's why you see 3 treatment arms in addition to a control. So in addition to, obviously a product that has a clinically meaningful difference to patients, we also want to ensure we can scale up this product and provide it commercially. So this slide walks through our process. And as I mentioned earlier, we are an off-the-shelf product. And what we do is we start with pooled qualified cord blood units that we procure from public cord blood banks. So all of these units have been screened for various infections. We isolate out the CD34 cells from those pooled units. We introduce them to our platform where they self-renew in the case of dilanubicel, and then they are expanded 250 to 300-fold. And it's that product then that is aliquoted into fixed doses of dilanubicel, either 800 million CD34 cells, 300 million, or 100 million. In the transplant setting, we anticipate launching with an adult dose of 800 million CD34 cells, and a pediatric dose will likely be 300. In AML, again, we're looking to define what dose that will be. The key point, though, is that all of this is manufacturing that's done ahead of time and inventoried so that when a physician decides to treat a patient, the product can be shipped on demand directly to those treatment centers. In terms of scaling our process, we initially started this process at Fred Hutch. In that manufacturing process, we used a single donor. But early last year, we transferred from the Fred Hutch to UC Davis, where we began to pool our cord blood products. And this product is actually the product that's being used in our AML launch trial. Because of the dialogue we've had with some of the regulatory authorities and the potential that we'll be at minimum a phase 3 company and potentially a pre-commercial company next year, we engaged WuXi AppTec in Philadelphia as our GMP facility that'll support the phase 3 and commercial supply. And then at some point, I think we all want to control our own manufacturing, and that's something that we'll look to do downstream. So in terms of how we're preparing for commercial success, when we look at this initial transplant market, we know that this is a well-organized and concentrated group of treating physicians. There are about 60 transplant centers that account for about 80% of use. And these centers oftentimes are referred to or designated as centers of excellence. In research we've done with payers, we've learned that payers direct patients to these centers of excellence, and they maintain their designation by improving patient outcomes. So as we build our value proposition and we look at how are we offsetting costs associated with that transplant, in addition, how are we improving patient outcomes as well? So that's the valuation model that we're building, and that obviously is work that we'll continue to do as we build our market access strategy, both here in the US and in Europe. So to summarize the near-term opportunity, you can see in the US, we're looking at a market of about $1.5 billion. We do have orphan and fast-track designation there. In Europe, we've had formal scientific advice that indicates with positive data from our cord blood study, there's an opportunity for accelerated approval there. We do have prime designation and orphan designation there. And in Japan, for those of you who went to the sessions yesterday morning, we have talked to the PMDA with the intent of capitalizing on the regenerative medicine initiative that they have there. We've completed our formal meetings with our non-clinical and quality colleagues, and we now are in the process of determining the small studies we need to do in Japan to support submission for a time-limited approval there. So this is our beauty shots. So this is our leadership team, and I just want to say I think everyone probably is very proud of their leadership team. I am very proud of this team that we've built. Everyone here is an expert in their field, and I specifically want to call out Colleen Delaney, who's the founder of Nohla and currently our chief scientific officer, and she really has been spending this last year building up our internal research team and our programs, because in addition to our near-term opportunities, we also, as a platform company, are progressing other programs forward from that platform. So milestones, we've achieved a number of regulatory milestones this year. We do, 2019 is a pivotal year for us as we look to read out our top-line data in cord blood transplant and our AML data in intensive chemotherapy later that year. And then finally, I just want to ensure that we acknowledge our patients. They really are the anchor of what we do, and without their participation in the trials and their courage, we wouldn't be here. So, 0 minutes, wow. Okay, thank you very much.